I feel like usually when we picture a cell, we just think of this microscopic water balloon, like just a flimsy, flexible plastic bag holding a bunch of biological goo that's all just floating around together.
Right, yeah.
Yeah.
A very fragile, very simple boundary. Basically just there to keep the inside in and the outside out.
But I want you to completely erase that image from your mind. Just throw it out. Instead, imagine a cell's membrane as a massive, heavily guarded, two-sided fortress wall.
Oh, I like that analogy.
Yeah, so there is an inside facing the courtyard, right, and an outside facing the battlefield. And the guards standing on this wall are just obsessively dedicated to keeping certain things strictly on the inside.
It really is a highly militarized zone, structurally speaking. And, the architecture of that wall actually dictates whether the cell lives or dies.
Which is wild. And what happens when that fortress wall starts to break down, specifically in our brains? Well, that is exactly what we are exploring today. We have an incredibly fascinating source to guide us for this. It's a 2026 paper published in the journal Cell, Death, and Disease.
Right, and the mission of this deep dive today is to really explore this profound plot twist in cellular biology.
It really is a plot twist.
It is. We are looking at a brand new discovery about how mature brain cells, our neurons, actually die. And how a tiny protein that scientists thought was just, a simple little machine is actually a critical structural pillar keeping our neurons alive.
Okay, let's unpack this. Because to really appreciate the plot twist, we first have to understand the normal, everyday state of that fortress wall.
Exactly. So the cell membrane isn't just one single layer of fat. It is a bilayer.
Right, 2 layers.
Yeah, 2 layers. It has an inner leaflet facing the internal cytoplasm of the cell and an outer leaflet facing the extracellular space. And this membrane is highly asymmetrical, like it's not a uniform mixture at all.
Meaning certain fats or lipids belong exclusively on the outside and others belong strictly on the inside.
Precisely. And the absolute star of our show today is a very specific lipid called phosphatidylserine, or we can just call it PS for short.
PS, right. And in a healthy cell, PS is kept on a very tight leash.
Extremely tight. It is strictly confined to the inner leaflet facing the courtyard. But when a normal cell becomes irreparably damaged and enters programmed cell death, which is a biological process we call apoptosis, the cell essentially just throws in the towel.
Right.
It takes that PS and flips it from the inner leaflet to the outer leaflet.
I always picture this like putting your trash cans out on the curb. Flipping PS to the outside of the cell is literally an eat me signal.
Yeah, that's exactly what it is.
It tells the immune system's roaming garbage trucks, these cells called phagocytes or microglia, to come along, swallow the dying cell whole, and dismantle it. So trash on the curb means the house is slated for demolition.
Which is how it works in almost every single cell in your body. But what the researchers found here is a massive biological anomaly.
Look at the twist.
Yeah, in mature neurons, flipping those PS trash cans to the curb isn't necessarily a death sentence. They observed that it can actually happen during non-apoptotic stress.
Wait, meaning the cell isn't intentionally initiating a programmed suicide sequence, it's just under intense, overwhelming stress.
Precisely. It's just stressed. And even more surprisingly, this lipid flip in neurons can be reversed.
Wow.
The neuron can literally pull the trash cans back inside the house if it survives the stress.
That challenges basically everything we learn in entry-level biology. I mean, it's wild. But, you might be wondering how scientists can actually see a microscopic lipid flipping back and forth in real time.
It's not easy.
No, it's not. To watch this happen live, the researchers use this brilliantly engineered tool called a PSIVA probe. It is a polarity-sensitive fluorescent probe that you just drop onto living cells in a petri dish.
The mechanics of PSAV are actually quite elegant. When this probe is just, floating around in the watery environment outside the cell, it stays completely dark.
Nothing happens.
All right, nothing. But if a cell flips its PS to the outside, the PSAVA probe binds to that specific lipid environment.
And when it binds, the actual physical molecular structure of the probe twists and changes shape, right?
Exactly. That structural change allows it to absorb and emit light. So it suddenly fluoresces bright green under the microscope.
And if the neuron manages to recover and pull the PS back inside,
then the probe gets knocked off, it goes back into the water, and goes completely dark again.
It is exactly like a motion sensor spotlight that only turns on when the trash is exposed on the curb.
That's a great way to put it. And by watching these spotlights flicker on and off under high-powered microscopes, the researchers realized that mature neurons are playing by a completely different set of rules when it comes to membrane asymmetry and survival.
Okay, so let's do a quick recap here just to make sure we're all on the same page.
Good idea.
So, Bullet 1, we've got this lipid, PS, that's normally locked inside the cell. Bullet 2, usually flipping it to the outside is a signal for the immune system to devour the cell. But bullet 3, mature neurons can somehow flip it to the outside during intense stress and pull it back in without dying.
That's the core of it, yeah.
So, if you are listening to this, I want you to think about the implications of that for a second. Have you ever considered that the physiological signals your cells use for death might actually sometimes just be desperate cries for help?
And that perspective is exactly what drove the researchers to the next phase of their experiment. Because if PS flipping is a cry for help, well, they needed to pinpoint exactly what kind of stress triggers it and where that stress actually manifests.
Right. They needed to simulate different environments in the lab. So they set up 2 distinct scenarios. First, they wanted to simulate a normal, heavy workout for the brain.
Yeah, some healthy exercise.
Exactly. To do this, they used a chemical called bicuculin, or BIC for short. Adding BIC basically puts the neurons on a treadmill. It enhances normal synaptic activity and makes them fire off action potentials at a high but totally healthy rate.
And the second scenario was designed to be a toxic event. For this one, they bathed the neurons in a compound called NMDA. They were essentially flooding them with high levels of glutamate. This simulates a severe pathological condition known as excitotoxicity.
Excitotoxicity. I mean, just saying that word, it practically sounds like an alarm bell going off.
It really should. It's one of the most destructive forces in neurology.
Yeah.
It is what happens in the brain during a stroke or after a traumatic brain injury, or even chronically in neurodegenerative diseases like Alzheimer's.
So what is actually happening physically during excitotoxicity?
Well, what happens is the extrasynaptic receptors on the neuron get completely flooded by neurotransmitters that just won't dissipate.
Like the signal gets stuck on.
Right. The channels get locked open and massive, completely unregulated amounts of calcium rush into the cell. And this calcium flood just overwhelms the internal systems. It ends up activating enzymes that literally start chewing up the cell from the inside out.
Okay, so they have these 2 groups of neurons, the ones getting a healthy workout with BIC and the ones drowning in the toxic NMDA flood.
Right.
So they turn on the PSIVA spotlights. When the neurons were just exercising normally, the spotlights stayed dark, the lipid wall held strong, no PS flipped to the outside.
Completely normal.
But when they hit them with the NMDA excitotoxicity boom, the microscopes lit up with massive, sustained green fluorescence. The PS was flipping to the outside in droves.
And this is where the researchers had to be incredibly rigorous. Because, when you see new molecules appearing on the outside of a cell, you always have to ask, did the cell just build those from scratch?
Oh, like manufacturing new trash to put on the curb?
Exactly. So to prove it was actually a flipping mechanism, they utilized lipidomics. It's a mass spectrometry analysis.
Right, which basically just weighs and counts the molecules in a sample.
Exactly. And they found that the total pool of PS in the cell remained exactly the same before and after the toxic flood.
Wow, so the cell wasn't manufacturing brand new PS, it was literally taking the lipids it already had and forcing them from the inner leaflet to the outer leaflet.
Exactly. But here is where it gets really interesting. They didn't just look at the whole cell. They zoomed the microscopes in incredibly close on the dendrites.
Those are the long, delicate, tree-like branches that neurons use to receive signals from other cells, right?
Yep. And they noticed that the PS wasn't just popping out uniformly all over the membrane. It was clustering in highly specific, organized hotspots.
Hotspots.
Yeah. And these hotspots were located almost entirely at dendritic branching points and dendritic spines. Basically, the actual architectural joints of the neuron.
It's a crucial detail that really shifts how we view cellular damage, right? Because the exposure isn't random. It is spatially organized at very specific subdomains of the membrane.
It is.
I look at this kind of like a suspension bridge under heavy traffic stress. When a bridge is overloaded by too much weight, the concrete doesn't just crack perfectly evenly across the entire surface.
No, of course not.
The stress fractures accumulate at the mechanical joints, the branching points, the load-bearing pillars. But I do have a question about this.
Sure.
When they hit these neurons with that toxic NMDA flood, did the toxicity physically create new vulnerable spots on the membrane, or did it just exploit the structural weaknesses that were already there?
That is exactly what the researchers wanted to know. And the data points to something profound about early neurodegeneration. The NMDA toxicity did not increase the number of hotspots.
It didn't.
No, it didn't create new cracks in the bridge. Instead, It intensely concentrated the PS exposure at those pre-existing architectural junctions.
Ah.
The stress basically exploits the structural domains that are already doing the heavy lifting, which points to a localized vulnerability right where the membrane interacts with the cell's internal skeleton.
Okay, so let's do another quick recap, pulling this all together. Normal brain activity leaves the structural integrity of the cell alone, but toxic overstimulation causes a massive disruption, flipping lipids to the outside, specifically at the weakest structural joints of the neuron's branches.
That's a great summary.
So think about the branching points in your own life or work for a second, whether it's a schedule, a team, or a piece of software. It makes you realize that the junctions are always where the stress fractures first, doesn't it?
Absolutely. The architecture really dictates the vulnerability. So now the researchers knew exactly where the PS was flipping, and they knew what specific stress triggered it. But that begs the fundamental mechanical question.
Right. Who is physically moving these lipids?
Exactly. If the trash is mysteriously ending up on the curb during a crisis, who is physically moving the cans?
So to figure this out, they set up what is essentially a biological police lineup, right? Because in our cells, there are nearly 20 different specialized enzymes responsible for moving lipids around the membrane.
Yeah, quite a few suspects.
You have flippases, which use energy to move things from the outside in. You have floppases, which move things from the inside out. And scramblases, which just mix things up in both directions.
Right. And the researchers screened 13 of the most likely suspects in these mature neurons to see which of these enzymes were being affected by the toxic NMDA flood.
And what did they find?
Well, the results of that screening were remarkably exclusive. Out of all 13 candidates, only one single enzyme's expression levels dropped significantly during the toxic stress.
Just one.
Just one. It was a specific flippase called ATPA2.
The prime suspect.
Without a doubt, when the toxic NMDA hit the cell, both the mRNA blueprints and the physical protein levels of ATPA2 just plummeted. It was being destroyed.
But what is really telling here is what happened to its closest relative, right? An enzyme called ATP81.
Yes, this is fascinating. From a biological standpoint, ATP81 is almost identical to ATPA2. They are closely related cousins. Yet during this massive toxic event that basically obliterated ATP82, the levels of ATP81 remained completely unchanged.
It was totally unaffected.
Totally fine.
Now, if you're listening and you're a bit skeptical, you might be thinking, okay, that's an interesting chemical reaction in a plastic dish in a laboratory.
Right, in vitro versus in vivo.
Exactly. But the researchers took this out of the Petri dish and proved it in living, breathing animals. They looked at 3 entirely different mouse models of severe neurodegeneration to ensure this wasn't just some fluke.
And those models are really important.
Definitely. First, they injected NMDA directly into the retinas of mice, which perfectly mimics the damage caused by glaucoma. Second, they used catenic acid to induce seizures, purposefully damaging the hippocampus to model epilepsy. And third, they looked at transgenic mice, the APP-PS1 models, that are genetically engineered to develop human-like Alzheimer's disease.
3 totally different conditions.
Yeah. And in all 3 live models, across completely different brain regions and totally different diseases, the exact same mechanism unfolded. ATPA2 levels crashed, and ATPA1 was completely fine.
It's remarkably consistent.
But let me push back on this because it honestly seems counterintuitive to me.
Oh, how so?
Well, if ATPA1 and ATPA2 are basically identical cousins and they live in the exact same cell membrane, why does neurodegeneration only take out ATPA2? And maybe more importantly, if ATPA2 goes down, why can't ATPA1 just step up and do the job? Doesn't evolution usually provide a spare tire? Why can't the backup system just pick up the slack?
It is a really great question, and it directly challenges a long-held assumption in biology about redundancy.
Okay.
For decades, scientists assumed that because these 2 flippases were so similar, they simply covered for each other. If one failed, the other would just work a little harder.
Makes sense on paper.
Right. But this paper definitively proves they are not functionally redundant in mature neurons. Through evolution, ATPA2 has taken on a unique, highly specialized, and completely irreplaceable role that ATPA1 simply cannot perform.
Wow.
When ATPA2 goes down, the whole neuronal system feels it, and the backup, ATPA1, just sits there, incapable of stopping the collapse.
Okay, time for another recap. So we know that out of all the lipid-moving enzymes, only ATPA2A is destroyed during neurotoxic stress. We know this holds true across live models of glaucoma, epilepsy, and Alzheimer's. And we know the backup copy can't save the day.
Spot on.
Which leaves us looking at this failure cascade. Listeners, think about this. If a system has a built-in backup, but a critical failure still brings the whole thing down. It usually means we misunderstand what the primary system is actually doing.
Precisely. It means the primary system is doing a second job the backup wasn't built to do. Which brings us to the absolute climax of this paper.
The big reveal.
Yeah. The researchers needed to know why the loss of this specific enzyme, ATPA2, is so devastating to the brain cell. Does the neuron die simply because ATPA2 isn't there to pump lipids back inside? Or does it die because the physical presence of the ATPA2 protein is suddenly missing from the fortress wall?
Oh, I see. This is the difference between losing the activity of a machine versus losing the physical scaffolding of the machine itself. So to test this, the researchers used two incredibly clever genetic and molecular methods. Method one was basically the biological wrecking ball. They used a tool called shRNA.
Right. shRNA stands for short hairpin RNA.
Yeah.
And it acts essentially as a genetic sensor, or like an interceptor.
So before the ATPA2 protein can even be built at the cellular factory, the ribosome, the shRNA, intercepts the genetic blueprint, the mRNA, and just shreds it.
It destroys it.
So the cell stops making the protein entirely. There is a literal physical hole in the cell membrane where ATPA2 used to sit.
And the results of method one were completely catastrophic for the neuron.
What happened?
When the protein was physically removed from the wall, the dendrites physically shrank and lost all their intricate complexity. The axons degraded. They showed massive clusters of a stress marker called SMI32, which basically indicates severe structural failure.
Ouch.
And ultimately, the cell triggered the activation of cleaved caspase 3.
And cleaved caspase 3 is essentially the executioner enzyme. Once that is activated, it is the biological point of no return for cell death. The cell is actively dismantling itself, so the neurons died off rapidly.
Very rapidly. So destroying the physical machine completely kills the cell. But what about method two?
Right.
For this, they didn't use a wrecking ball to destroy the protein. They used a highly precise wrench just to jam the gears of the pump. They designed a custom-built peptide called PI8A2.
Okay, to understand how this tiny wrench works, we really have to look at the 3D structure of ATPA2. For this flippase to actually pump lipids, it can't work alone. It needs a cofactor, a little buddy protein called CDC50A, to bind to specific outside loops on its structure.
Right. And the researchers analyzed the exact 3D contact points where these two proteins basically hug, and they built the PI8A2 peptide to perfectly mimic that binding site.
So it acts as competitive binding.
Yes. When they add the peptide, it plugs the hole where the cofactor is supposed to attach. It completely jams the flipping action, preventing the enzyme from pumping any lipids whatsoever.
But, and I want everyone to listen closely because this is the absolute most crucial part of the whole deep dive. The physical ATP8A2 protein is still sitting perfectly intact in the cell membrane. The building is still standing. The doors are just super glued shut. It can't pump.
Exactly.
So what happened? Well, when they just jammed the machine with the peptide, the PS trash cans flipped to the outside because the flippase was broken and couldn't pull them back in. The membrane asymmetry was totally ruined. But UT, the physical structure of the cell, stayed perfectly healthy.
It is amazing.
The dendrites didn't shrink, the axons didn't degrade into those SMI32 clusters, and the cell lived happily for days even though its lipid pump was completely broken.
Which is just, if we pull back and look at what this means, it is a monumental paradigm shift in how we understand neurobiology. It proves that ATP8 is not just a lipid-flipping machine. It is a vital structural scaffold. It is literally a load-bearing pillar in the membrane. By reanalyzing interactome datasets, which map how different proteins touch each other, they found that ATP8 physically anchors the outer membrane to the cell's internal skeleton.
Wow.
Yeah, it binds specifically to heavy-duty structural proteins like spectrin, ankyrin 2, and CAMKII.
Okay, so when excitotoxicity destroys ATPA2, it's not just breaking a lipid pump. It's physically knocking out the load-bearing pillars at the branching joints of the neuron. The bridge collapses because the steel cables literally snapped.
Exactly. The lipid flipping activity is basically just its day job. Its actual physical existence in the membrane is what holds the neuron's architecture together.
Let's quickly recap this climax because it's so important. The takeaway here isn't just about a broken pump. Destroying the protein entirely causes the neuron structure to collapse and die. But if you just jam the pumping action, leaving the physical structure intact, the cell survives. ATPA2 is acting as a physical anchor. So, to you listening, think about what happens when we misunderstand the fundamental purpose of a tool. We thought this whole time we were looking at a tiny pump, but it turns out we were looking at the foundation of the house itself.
And recognizing the true purpose of that tool fundamentally changes how we try to fix it when things go wrong in human disease.
So what does this all mean for us? How do we take this incredibly complex, microscopic plot twist and actually use it to help people suffering from neurodegeneration?
Well, the researchers took the final logical step. If losing the structural pillar causes the house to collapse during toxic stress, what happens if we reinforce it before the storm hits?
Oh, like boarding up the windows.
Exactly. They use a specialized gene construct to overexpress IP882 in mature neurons. They essentially hijack the cell's machinery to pump extra massive amounts of the IP82 protein into the cell membranes, massively fortifying the fortress walls.
And the results were like giving the neurons a biological super shield, right?
Oh, totally. Not only did these supercharged neurons keep their lipid asymmetry perfectly balanced under normal conditions, but when the researchers blasted them with that toxic NMDA flood, they survived.
Incredible.
They became incredibly resistant to excitotoxic death.
So this is where the therapeutic shift happens, and why this paper is so important for the future of medicine. Traditionally, pharmaceutical companies looking at enzymes will try to design drugs that activate them.
Right, agonists.
Yeah, agonists that make the pump work faster or harder to clear out the problem. But this paper proves that for devastating diseases like Alzheimer's, glaucoma, or epilepsy, boosting the pumping activity isn't going to save the cell.
No, it won't. Because if the toxic stress is physically knocking down the pillars, turning the remaining pumps up to maximum power won't stop the ceiling from caving in on you.
It's structural.
Exactly.
Yeah.
We need a whole new class of therapies that protect the expression of AP2. We need drugs that intercept the toxic signals before they destroy the physical protein, or advanced gene therapies that continuously supply the neuron with fresh structural pillars, because its mere physical presence in the membrane is the only thing keeping the brain cell from structurally collapsing.
Yeah. I mean, we started this deep dive looking at what we assumed was a simple, flimsy water balloon, and we ended up uncovering a microscopic fortress where the garbage disposals are actually holding up the roof.
It's quite the journey.
It really is. I want to thank you for joining us to explore this incredible biological puzzle. It completely changes how we view cell death, neurodegenerative disease, and just the hidden architecture of our own brains.
It really does shift the entire perspective. And, as we wrap up, I want to leave you all with one final thought to mull over.
Lay it on us.
If a simple enzyme like AP2 is secretly acting as a hidden, load-bearing scaffold for the cell membrane, what other simple machines or basic functions in our bodies, or even in the technology and organizational systems we build every day, are secretly serving as the structural pillars holding everything together without us even knowing?
Man, that is something to think about the next time you look at a bridge, or a complicated system at work, or even just a diagram of a brain cell. Keep questioning the world around you. Keep looking for those hidden structures, and we will catch you on the next deep dive.